So I think next talk is by Dr. Panigoro. The title is the Variance Profiling of BRCA1/2 genes through next generation sequencing in young women with breast cancer. So can you start?
Hello everyone, my name is Afika Indaparamita. I'm from the Department of Medical Chemistry and Bioinformatics Professor Disciplinary, Faculty of Medicine, University of Indonesia. So today I would like to present our research about variance profiling of BRCA1 and BRCA2 genes through next generation sequencing in young women with breast cancers. Okay, talking about genomic sequencing, nowadays the most used platform is next generation sequencing. And one of the application from NGS is to help to identify cancer-related variants that we did in this study. BRCA1 and BRCA2 are human genes that produce tumor suppressor protein, and this protein helps repair damaged DNA. If either of these genes is mutated or altered, DNA damage may not be repaired properly. As a result, cells are more likely to develop additional genetic alteration that can lead to cancer. So in this study, we aim to identify the BRCA1 and 2 mutation using NGS-based technique in breast cancer patients undergoing treatment in several hospitals in Jakarta. And here's our subjects. We got 75 women patient under 45 years old, and all of the patient already given the informed consent. And this study was already approved by Ethical Committee at Faculty of Medicine, Universitas Indonesia on October 16, 2017. For the library preparation, already described in our previous data article that already published in Data in Brief, volume 32 on October 20 and 20. First step, we did a DNA extraction using, we extracted DNA from Buffy code using reagent from KM DNA minikit. And then we did a library preparation using target-rich hereditary cancer panel from Calyx Genetics. And then we sequenced using X6500 platform. And some of the raw vascular file already storage in SRA in NCBI. Okay, next step is bioinformatic analysis. We did from QC of the NGS auto file FASTQ format until digital visualization using several software, as mentioned in slides right now. And then this is the patient characteristics. The mean of age of onset is 34 years old. And the patient that have breast cancer family history is 18 or 24% from our samples. And this is the variance that we got from BRCA1 and BRCA2 genes. We visualize using lollipop plot. In here, we 3 different colors dots. That means the green one is a missense mutation. The black dots in here is for truncated mutation, which is a nonsense, friendship, or splice site mutation, and the pink one is for synonymous variants. In BRCA1, we got 1 truncated mutation in here. That annotate InCoB database as a loss of function variants, and also 1 missense variants that also annotated as a loss of function variants. In BRCA2, we got 4 truncating mutation, and this is 1, 2, 3, and 4 mutation. That all of the mutation already are annotated as a loss of function variant from InCoB database. This is the result. The pathogenic, likely pathogenic, and fused variants or variants of uncertain significance in BRCA1. And here we see that one of the patients that have family history of breast cancer have pathogenic variants. And we see here in BRCA2, we got 4 pathogenic variants, which 3 of them has a breast cancer family history. So in 22.2% of pathogenic variants detected in patients with breast cancer family history. And in sporadic breast cancer patients, it's only 3.5%. Family history of the disease, it means in breast cancer, can increase a woman's risk for having this disease, having have a younger age at diagnosis or onset of diagnosis, and have a higher frequency of bilateral disease than those with sporadic breast cancers. So in summary, we found interference, silent, nonsense, missense, and frameshift mutation in our BRCA1 and BRCA2 genes. And the pathologic variants of BRCA genes were detected higher in a patient with familial history than in sporadic ones. So thank you very much for your attention. Bye. Hello everyone.
Thank you for your nice talk. So, are there any questions from audience? No question at all. Yeah. So, so I have a question. So, you made analyze the BRCA1 and BRCA2 loci by next generation sequencing, and can you identify several so validation of your results? So maybe yes. So can you compare with can you compare your result with the other so studies in women's breast cancer or something like that? So
Yeah. In several variants, we already it's already available in the database, but some of them we found it's novel. So if I'm not really get about meaning of the validation, but from some variants, yeah, we found it also in database. So it means another research already found the same variants that we got in our samples.
So maybe you can look at the TCGA or some other cancer database. So maybe so you may get some your own result from the your original result compared with that kind of database. So any other questions? Uh, Maybe was there any integration between genome data and other clinical progenitor factor what to determine the diagnostic predictors? So, uh, so have you already integrate your so sequence result with the clinical some clinical results?
Yeah.
Maybe
Okay.
yeah.
Actually, we already did the like correlation with genome data and clinical factors, but in this lightning talks, I didn't show it yet, but
Okay.
yeah, we already did the analysis in maybe in our previous publication. But yeah,
Okay.
we did it.
Okay. Thank you.
Yeah, thank you very much.
Okay. Thank you very much.